Thanks so much for being on. I'm going to need the pointer. Maybe I will. Oh, good. Okay. Thanks for having me back. It's always a bit of a surprise. So I'm Mark. I'm a psychiatrist from across the road at the Royal Melbourne Hospital, where I've been seeing patients with NPC for about the last 25 years. Since we first saw our first patient over there in the year 2000, we're an adult unit, the neuropsychiatry unit, and so we see people with complex, younger people with neurological conditions that often have psychiatric sequelae, and that's how I first saw my first NPC patient. And I think, as we were discussing before, once you at least are familiar with the disease, that enables readier diagnosis, and I think that's important. And I think we've been quite good at diagnosing patients here in Australia, particularly with adult disease. So I've been asked, when Dee asked me to talk about this, I thought, God, I'm not sure I'm going to have that much to talk about in comparison to last year. You know, how much has really happened? Everything feels a bit slower after COVID. We're all a bit exhausted. And I started talking to some colleagues saying, you know, that thing you were doing, that thing, do you reckon you've got any slides you could give me about that thing? And so suddenly, like in the last 24 hours, I've got all these slides. So we've got a little bit to talk about, but we will talk about the fact that we've got a great patient population in Australia who've worked so closely with us. It's always been a great example of how people work together. In the last 20 years, we've been able to put out 49 different research papers on NPC, and hopefully some of the stuff you'll see today will be paper number 50. Since I spoke last year, I thought I'd talk a little bit about just a couple of the other papers that we've put together. 3 have been published. This one, people will know who've been involved. We do a lot of scanning of our patients. My initial doctoral work was in MRI scanning in major mental illness. So we've often used some of those techniques on our NPC patient population. This was just really to show that midline brain structures with a Japanese collaborator, Satomu, looking at midline brain structures. People know I'm very interested in how disease affects the NPC patients' brains. This is just about midline brain structures, showing that in a way, some of the key findings that we see, and this actually come from a lot of the schizophrenia work that we've done. This aligns a little bit with schizophrenia, but it also suggests that some of the brain changes that we see, even in our adult patients, were present very, very early on in life and even before birth. Some people know about this paper, N-acetyl-L-leucine, now a really nice paper from the clinical trial that was published earlier this year in the New England Journal. So there's a lot of trial activity that happens in Australia. It's been great to be involved. I'm involved in a number of other neurodegenerative disorders, but really NPC was the first that I was involved with that had an illness-modifying treatment that was available when we first started using miglustat way back in 2008. One of the other things that we're looking to do, particularly, is looking at plasma biomarkers. NFL is not just a sport, it's also a key cytoskeletal protein in axons. And NFL is a really interesting protein that you could only historically detect in the cerebrospinal fluid, the fluid bathing the brain, but now with new machines, we can actually detect it in the blood. And it looks like it's a really interesting biomarker. In an early study of ours, we've shown that NFL is quite different in our NPC patients. There are higher levels suggesting some active brain changes and brain processes are going on. The glial fibrillary acid protein, which is often a marker of astrocytosis, is not particularly elevated. We have a couple of other interesting plasma studies coming up. So this is the bit where I asked people to tell me about those things that they were doing. So this is, again, people will have some idea that we've been doing a lot of neuroimaging work in NPC and published some of the first group neuroimaging data where we see, well, this brain region's a bit different, this brain region's a bit different in people with established illness. But one of our young psychiatrists is interested in really brain network-based statistics. And this is really working out how well the different parts of the brain communicate with each other. So it's another layer above. He's given me some really difficult slides to talk through. So these are not going to mean that much. But really what this is, it looks at a particular technique called DTI, which looks at the connecting fibers of the brain. I wish you'd given me some DTI slides to show you what it means. But basically, it's about kind of how different brain regions talk to each other, how well connected they are when you compare a group of patients versus controls. And there are all sorts of ways that you can look at connectivity across brains. And he's given me about a dozen, which I'm not going to talk through. I'll take you, in the interest of time, to... So you can see here, we can, when we get an MRI scan, there's a reason MRI, it was a Nobel Prize winning technology about the way you can make protons dance in a magnetic field. We can segment the brain into all of these different regions and look at how there are different degrees of connectivity between these regions. Cortical regions here, these are the outer part of the brain. Subcortical regions here. One of the really nice things is what we see with the connectivity is it maps really nicely to a paper we published almost 15 years ago in neurology, showing the subcortical changes that we see in NPC. And these are, different scanners, different subjects, etc. But really the key slide is this one. Now, if you imagine this is we're looking at the brain from underneath. So right is left. So this is the left hemisphere, this is the right hemisphere. This really shows the differences in NPC patients to controls about the kind of the global connectivity, but regional connectivity between different brain regions, between cortical brain regions and each other, and cortical brain regions and subcortical brain regions. And it is interesting because it tells you that it's predominantly a left frontotemporal and left frontal subcortical change. Why is that interesting? I see adult patients, about 40% of our young adult patients have a primary psychotic disorder that looks very much like schizophrenia. This is why I got very, very interested in what was happening with our NPC patients and how we could apply our schizophrenia imaging techniques. So it kind of confirms that might be underpinning some of the psychiatric illness we do see in younger patients. Some, not all, about 40%. One of the other interesting things for us is because we often treat young patients with neurodegenerative disorders. We have a very large Huntington's disease program. We also have a large young onset Alzheimer's disease program. I was talking to someone before about younger patients with Alzheimer's. So these are people in their 20s, 30s, 40s with Alzheimer's disease. And getting a sense of what are the key regions of change. We know that in NPC1, and again, we've been doing formal neuropsychological testing, so really in-depth 2 to 3 hour neuropsychological testing on our patients now for a very long time. We know that we see things that you would see, particularly as families, kind of day-to-day living, impaired attention span, concentration is impaired, multitasking. We all know about reduced fine motor skills in particular, memory, slower to learn new information, recall is impaired, but particularly in executive functioning, things like inhibitory control, being able to have that filter, the brake line seem a little bit more faulty, planning, organisation, being flexible and getting started. And we know that these are largely driven by, this is kind of a little bit pixelated, by the brain regions where we see the most change in NPC. We know that we see that a lot in the cerebellum, but the cerebellum doesn't just control fine motor functioning, it has a key role in modulating executive functions, higher cortical functions as well. And look, this is going to vary across different age groups. So when we see children, cognitively, they will get delays in their developmental milestones. We've heard a little bit about that today with pediatric patients, learning difficulties, and Chris has also mentioned that kind of deviation in there, that trajectory, what's expected and then what occurs, and a significantly increased need for support. In adults, there's changes in mood, mental state, behaviour, executive impairment in particular. We will sometimes see in our later onset patients very subtle illness, and it's usually executive impairment, judgment, planning, initiation, inhibition that occurs first. So what we decided to do was look at all the testing we've ever done with all of our patients. And you can see this dates right back to 2001. We basically looked at a 20-year span of what was happening with our patients. It's a huge amount of data, lots of holes in that database, trying to kind of, get it clean and tidy to kind of pull the signal from that. It was great work by Michelle Chu, who's one of our PhD students. So she standardized all of this to normative data, particularly in terms of Z scores, and gave impairment as 2 standard deviations below the normative mean. We've done a lot of analysis of these, and these were the things that we pulled out that most patients had. different clinicians will choose different tests, different tasks, different times. There'll be the version 3, the version 4, etc. So what did we find? So we found that, and this is probably not a great surprise, that psychomotor speed is probably the biggest. It's impaired in most people, almost everyone. And remember, we see people who are minimally impaired across the spectrum. Executive function is the next big one. Language, spatial function, memory, recall. New learning, not as impaired as you might think, a little bit of a surprise for us. Here we see in the green, these are mostly motor speed measures, but then we see executive functioning using inhibition on the DCAFs, and then color naming on the Stroop. So these are two key measures of executive functioning. These measures here are very much affected by the motor function of patients. So they might have dystonia, ataxia, but really what it's telling you is the headline stuff is there are specific aspects of executive functioning that are often the first to change and present in more people. Tests with lower impairment rate, a little bit of a surprise. Some of the memory measures were actually not too bad. So it's interesting seeing adult patients because people progress, they have a later onset, they progress slowly, and you can often see what is, because that kind of cascade of changes isn't so collapsed that everything happens at once. You can kind of pass them out. That we often see those executive changes happening first. Why are these important? Because potentially they're useful illness biomarkers. They're targets for cognitive remediation. But I think the really important thing for us is if you know what are the key things, what's really sensitive to change, then you can develop a relatively targeted battery. Not everyone can withstand 3 or 4 hours of testing. But if you could condense that down to 30, 40 minutes, then that might be a lot more tolerable for people and yield just as much data. So we have been working to develop a blood test for dementia, if you like, for quite a while. There are many people who are doing very similar things. I presented a little bit about this before. I think this is the QR code for the MIND study. We have been looking at biomarkers, particularly with collaborators in Gothenburg for quite a while in a range of different disorders, young onset dementias, but other disorders from delirium, post-anesthetic patients, footballers, etc., for quite a while. This was our study looking at NFL, so neurofilament light, there's neurofilament light, there's neurofilament heavy. These are important cytoskeletal components, particularly in axons. Some of our work previously, which I haven't shown today, has shown how much the white matter is affected, how much axonal structure can be affected in NPC. So when these break away, these are released into that fluid around the brain, and some of that gets into the blood. And with the advent of new machines like Simoa, we can detect much, much lower levels that circulate in the blood. And here we see in our control group of 25 patients that were age matched to our NPC cohort, 11 patients, we can see the significant increase. We did, however, not see one of the other key markers that's looked at, particularly in brain injury. GFAP was not elevated in the NPC patients. This again is potentially, I think, a really useful biomarker. Our first publication on this some years ago showed that elevated NFL could be reduced when people were treated with cyclodextrin. And I know that there are a lot of people looking at treatment effects on these blood biomarkers right now internationally. And for those people who have been involved, we will be doing the 2-year follow-up very, very soon. So this last one second last one. So this is looking at immune function. We have a collaborator, Ilya Voska-Boynik at the Peter MacCallum. We did this a couple of years ago. Some of the patients and their families will remember me taking blood and then sprinting it across the road to get it to their lab so they could look at fresh blood and look at immune functioning under the slide. The really good thing is there are 2 walkways between the Royal Melbourne and the Peter Mac that cross Parkville. And this was the first paper we got out of this in Blood in 2022. Daniela Castablanco, PhD student, did a huge amount of work on this. And this is to show you what we did. And I just literally put the last video of this on 5 minutes ago. So killer cells are kind of like, it's a little bit politically on the nose, but they're kind of like Russian spies. So they come up to you with a poison pen. If you're a cell and you're infected with a virus or you're a cancerous cell, they come up to you, they stab you with the poison pen, and then they slink off away. So I'll show you what that looks like. So green cells are killer cells. Up here you see, bang, he's been stabbed. he shrinks away, etc. So that's an interesting system. It involves a specific system called perforin, which is what Ilya's interested in. And the perforin system has to punch a hole in and then inject something from these full of granzyme, these cytotoxic granules that are stored in lysosomes. So lysosomes, we all know about lysosomal storage disorders. Lysosomes also are important in this system. Now here's what happens. Here you can see in an NPC, this was actually a mouse study. This was an NPC. You can see it's going up there. It's jamming with the pen, jamming. Nothing's happening. So that was, this is Ilya's slide. A lot of interesting detail on how the perforin system actually works. It's slightly different between these two cell types, the cytotoxic T lymphocytes and the natural killer cells. They jam in these secretory granzymes. Sorry, these are Ilya's slides.
Question.
Yes.
Online.
Yes.
Later? Yeah, I'm just letting you know.
Ripper. I love questions when I'm presenting other people's data. That's fantastic. What's really interesting is it was a very interesting finding. Why is that relevant? So one of the questions has been, could this mean that people with NPC might have an increased risk of some infections, some cancers, or immune dysfunction? The answer is, when we actually looked at this in humans, that you actually need a really severe deficit to get a severe enough deficit in this system. The answer is probably not. Some people know about the increased rates of inflammatory bowel disease in NPC like Crohn's. Might it have something to do with that? Potentially. And this is just really to show me, he expanded this quite nicely into other lysosomal storage disorders, Gaucher's disease, not that different. Sorry. But we were keen to look at a range of other lysosomal storage disorders. So one is Niemann-Pick type A and B, which is acid-sphingomyelinase deficiency. We looked at Woolman disease. I think I wrote down the headline findings of this. Basically, if it was suppressed, you would see a separation of these two lines. Woolman disease was relatively normal. It had higher degranulation, so probably not particularly abnormal. So we skip past Woolman disease, not super exciting. There's an elevation in LAMP1, which I've spoken to Ilya about, not super exciting. We looked at eye cell disease, which is another type of deficiency. And look, in the end, they didn't separate. Not super exciting. That was a little bit of a surprise, but it's actually refined our knowledge about what's actually happening to think that it might actually be really quite important what builds up. So what builds up in the lysosome and how it impairs this kind of killing cell function is actually quite super important. So we showed in NPA, so Niemann-Pick disease type A, that there is a reduction in cytotoxicity, so its capacity to kill those virus or cancer affected cells. It's reduced by about 50%, and it looks like there's a fusion issue. So these kind of bubbles full of poison have to get to the cell membrane and then get to the other side and fuse with the opposite cell membrane, and that's impaired. So there you can see that separation. That's what we start to see. We then looked at NPC2. Now, most of our NPC patients that we see are NPC1, NPC2. It's a different protein, a different kind of taxi in the cholesterol trafficking system, if you like. But really with, and by the way, I didn't mention right at the beginning, these are all CRISPR knockout mice. So knocking out NPC2, you actually see an 85% reduction in killing activity. Now, there's a comparator disease called, I've got to remember, hemophagocytic lymphohistiocytosis, which is an autoimmune disease, and that would be considered pathogenically reduced. And we were having a discussion before about why we see so much more interstitial lung disease in NPC2. So there's certainly a big change in the immune system, greater in NPC2 than NPC1 that you see with this. So we know that NPC1 deficient cytotoxic T lymphocytes have this reduced cytotoxicity. So we're expanding that project into lysosomal storage disorders. We've been able to get 2 good NHMRC grants to make that happen. And this is just hot off the press. Again, some people will remember, again, I would be taking people's blood and then running it across the road to the BMAC, to the Australian Genomic Resource Facility, where we were doing whole genome sequencing on our patients. And this was led by Eliatan Niktab, who's a fantastic young brain who thought he'd developed a way to look for genetic modifiers that needed about 99% less people than a traditional GWAS. And it's really quite interesting. We did this in Huntington's disease as well, and only in 30 patients we were able to replicate the main GWAS finding in Huntington's disease, which was FAN1 as the chief genetic modifier. So we applied Eli's methodology to some pediatric patients from Taiwan and also patients in Australia. Now again, the difficulty in presenting other people's talks. Once in the US, I had to present Eli's slides, which were 15 slides of math. And I said to the audience, I'm a psychiatrist. This is not what I do for a living. Please give me in advance. So I went by his notes. But basically, what this does is you construct this kind of environment of different genes that interact with their products, interact on a cellular level, and see if that's enriched in NPC1, which it certainly is. So what's really interesting is it homed in on a family known as the lichen family. And this was looking at yeast, mice, and our human whole genomic samples. And we saw that in the mouse, where only lichen 1 and lichen 2 were examined, that there was a significant impact of that on. So if you knocked it out, the NPC mouse declined much more quickly, had much more rapid neurodegeneration. And it looked like there were a couple of key spots on lichen 3 that seemed to determine in adult and pediatric NPC patients, your age of onset, 2 in particular. And I was talking to Andrew Munkaxi about this earlier today. I'm saying, Andrew, with knockout, it's straightforward, but these samples, they've got all sorts of different mutations. How do you know whether it's these particular mutations on lichen 3 versus different mutations that different patients have? And he gave me an answer that I didn't really understand, so I won't try and repeat it. But why is this interesting? Because if you find a genetic modifier, it's potentially a new druggable target. That's why this is interesting, and that's why it's important. So I've given you the Cook's tour of what's happened in the last 12 months in research into NPC. Some of that's clinical, some of that is a bit preclinical, but even the preclinical stuff has relied on our patients and our families working with us. So none of this would be possible without good communication and collaboration, and of course a lot of hard work between the clinicians and the researchers and the families, and it's all built on goodwill, a bit like the bridge between the Royal Melbourne and Peter Mac that I would have to run those samples, that data across. Unless we have those bridges between the patients and families, the researchers, the clinicians, and I think the pharmaceutical groups and the organisations that fund them, we don't really get anything done. But you can see what can happen in a year when you do. And that's the end of my talk. Thank you very much.